Good evening, ladies and gentlemen. Tonight, the lecture is in English, so I will also do the introduction in English. I'm Peter Rudolph, former member of the board of this society, and I'm very happy to welcome you to the lecture of tonight, which will be given by Professor Roberta Croce from the Vrije Universiteit in Amsterdam. Roberta comes from Italy originally. She studied chemistry in Padova, and then she did a PhD in Milan on plant biology to move on to Germany for a postdoc at the Max Planck Institute in Mülheim an der Ruhr. Then she came back to Italy to start to work in the National Research Institute on Biophysics. So you see already the change between the two. And in 2006, she came to Groningen. She's in fact one of our Rosalind Franklin Fellows. And she was also promoted to associate professor here and got a VD grant. But then Amsterdam made her an offer she couldn't refuse. And this is how we lost her in 2011, but she has had a fantastic career ever since with the Vici and ERC Consolidator and Advanced Grant. Already in 2014, she was elected to the Royal Holland Society of Sciences and Humanities. And then in 2022, to the KNV, to the Royal Academy. And right now, she is the president of the International Society for Photosynthesis Research, and that is also what she is going to tell us about tonight. The floor is yours, Roberta.
Thank you very much, Petra, for the introduction and the invitation. It's a pleasure to be here, and it's always a pleasure for me to come back to Groningen. It's been a great period here. I really enjoyed it very much. This slide is coming. So what I'm going to talk about is photosynthesis. That is actually my passion, photosynthesis, harvesting the rainbow and beyond. So let's start from what you learned at the primary school, just to make sure that everybody knows what the photosynthesis is. Photosynthesis is the process that support practically all life on our planet. It's providing us with food and fuel, and the energy to make this food is, of course, light. What we want to do in my group is to understand this process more at the molecular level. And this is the story I will tell you a little bit of today. But apart from this, that I hope is very well known to all of you, maybe you also know that the photosynthesis not only is providing us with food and fuel, but also with the way to extract energy from them, because a secondary product of photosynthesis is actually oxygen. And so it's also the process that has changed completely the atmosphere of our beautiful planet. What you maybe can come with the surprise is that photosynthesis occurs everywhere. This is a movie that is been built by NASA. It's 20 years of recording from satellite. And what you see here, in fact, are the season changing and the changing in vegetation, for example, from brown, that is a zero photosynthesis, to a strong green, that is a lot of photosynthesis. So you're monitoring here photosynthesis. You can see it's happening everywhere, and this green is, of course, due to plants. You all know the plants are the major organisms doing photosynthesis on land. But actually, there is a lot of photosynthesis also in the sea. And if you go to purple here, to red, these are algae and cyanobacteria that are doing photosynthesis in water. And more than 50% of the primary productivity is actually happening in the oceans. And what according to me here is quite amazing is that these photosynthetic organisms, they live everywhere. And you know, light is the source of energy, and they are able to live in places where light is constant, 12 hours dark, 20 hours light, and where light instead changes a lot, and there are very long periods of dark. So this is also where temperatures are very low or very high. So this also tells you that photosynthetic organisms are actually able to adapt and live everywhere. And this is very good news because, of course, if you want to improve photosynthesis, we'll see in a moment, for an increase in crop productivity, with global changes and climate changes, we need also to learn how to adapt our plant, our crop. And yeah, there is a lot of information out of there that we can use. Now I will take you to my playground where photosynthesis really occurs at the molecular level. So we now enter in a leaf. And very soon you will see that there is a little hole in this leaf. It's called stomata, and there are cells that can open and close for gas exchange. Of course, the CO2 has to go in to be fixed into carbohydrates, and oxygen has to go out. Now enter into the leaf, then you see all these cells. These are the palisade cells, a lot of different layers of cell. We enter in one of them. You can see different organisms that you may recognize. The gray one here is the mitochondria, and the green here is the chloroplast. And the chloroplast is where photosynthesis occurs. We enter now in this chloroplast, and you see another system of membrane. These are the thylakoid membrane, and this is where the transformation of photon into electrons occurs. These are a stack membrane, many different stacks, filled with proteins. And all the proteins that are doing the transformation photons to electron are actually here. And this is what I'm interested in, and we are trying to understand how they work. You can also recognize several of them, for example, the ATP synthase and all the ATP going around. Okay, if I now look at this membrane in a more Canton way, these are the components of the photosynthetic system in these membranes. There are 4 multi-protein complexes that are participated in these reactions, and they are called here is photosystem 2. This is cytochrome B6F, this is photosystem 1, and this is the ATP synthase. I will talk about 2 of these complexes, photosystem 2 and photosystem 1, are full of chlorophylls. So they are the green one. They're responsible for the green. And what is happening there is that one of the many chlorophylls in this system is absorb a photon. Then the energy is transported to a particular chlorophyll here, where this is transformed into an electron, at least is used to do photochemistry. And then the electrons start to travel across the membrane. It's going out of photosystem 2, pass through cytochrome B6F, and go to photosystem 1, that in the meantime has done the same. And then is used for the reduction of NADP+ to NADPH. And if you can remember that this is the molecules that provide redox power to the metabolism. The only thing that is oxidized, that is missing an electron, is then one of the chlorophyll here, and in photosystem 2. And photosystem 2 is doing a fantastic chemistry, so is able to extract electrons from water. You can also remember that it's very difficult. You need a lot of energy to get electrons out of water, and the energy is coming from a light. So photosystem 2 is doing what is kind of a dream for many chemists, is using sunlight to, with a very high efficiency, take electrons out of water. Now, during this process, there is also, this is occurring in the membrane, so it's not permeable to protons. So there is an increase of the proton concentration on this side of the membrane. There is a proton gradient across this membrane, which is used by the ATP synthase for the synthesis of ATP. So at the end of these reactions, that are the one I'm very much interested in, we have ATP, this is the energy molecule of the cell, and NADPH with the reducing power for the cell. And these are then used for fixing CO2 into carbohydrates in what are called the dark reactions of photosynthesis. Now we go a little bit more in detail in this part. So this again is my membrane. This is a photosystem 2 complex, and now you see it seen from the top of a membrane. Now the different colors, they represent different proteins, and I also put them together. There are more than 40 of them, so you can see only a few of these. But you can see it's a modular system, and many of these proteins, they contain pigments. This, for example, LAC2, we'll talk about this later, is the most abundant membrane protein on Earth. It's responsible for more than 50% of the green that you're seeing outside, and contains this protein that contains pigments, and these pigments are chlorophylls, this tetrapyrrole kind of structure, and carotenoids. So this is beta-carotene. You're also very familiar to you because it's what you're eating with carrots. It's very healthy. So how do they work, this system? Now this is again a cartoon of the part of the photosystem 2. There are this in red here are the chlorophyll where they are able to do photochemistry, meaning when they are excited, then an electron can be taken away from one of them. And all the other chlorophylls here in the yellow part instead are the antennae, are the ones that are increasing the absorption cross-section of this one, so are getting more photons. And then they absorb light, and then they transfer the excitation energy to this chlorophyll where charge separation occurs. So how they work, then one is getting go to excited, and then the energy is transferred to this reaction center where charge separation occurs. Now, if we want this process to be efficient, is essentially that the transfer and the trapping of the photochemistry is very fast, because otherwise, of course, after you excite the chlorophyll, it will decay by natural processes. And this is actually what is happening. In all the processes that occurring inside this type of system are super fast. We go from 100 femtosecond from the transfer from one molecule to the next to 10 picosecond, 20 picosecond from one complex to the other. And the overall time, so the time from when one of the chlorophyll in the system is absorbing a photon to when charge separation occurs in the reaction center is about between 100 and 300–400 picoseconds. And all the natural decay instead are in the nanosecond regime. This means that all these systems are extremely efficient. Fundamentally, photosystem 1, that is one of the two systems that I introduced you before, is the best photon to energy converter, electron converter in nature with a quantum efficiency of 99%. So 99 out of the 100 photons that are absorbed by the system lead to charge separation in the reaction center. We want to study, we are studying this, and in my lab we are using what I call an integrated approach to study photosynthesis, because we are interested in the mechanism, but we want to study the mechanism in physiological relevant conditions. So this means that, of course, we extract protein and we study them in isolation, also at the single molecule level, but we are very much interested to study them in the complexity of the plant, so in vivo. And for this we need tools that allow us to find our way in the complexity of life. And we do this using a lot of molecular biology. We make mutants of our plants. We do a study by plant and algae physiology, so we study really the plant, the intact plant, and how they behave. We do biochemistry. We purify different complexes. We do a lot of spectroscopy, especially ultra-fast spectroscopy, femtosecond, picosecond, because these are the time regime that we are interested in when things happen in the membrane. And we combine this also with the modeling, molecular dynamics, and modeling for interpreting our complex measurements. And today I want to tell you two stories that are all about light, of course. The first one will be what is happening when our photosynthetic organisms are exposed to high light, when there is too much light. And the other is actually the opposite, what is happening and what we can do when there is actually very little light. Let's start with the high light. So high light is a problem. The plant, of course, use light as a form of energy, but they risk their life to do that. And I explain you with this cartoon. This is a representation of a chloroplast, and here are the light reaction. This is the stack membrane that I show you initially in the cartoon. So where all the light reaction occurs, as I told you before, at the end of the light reaction, we have ATP and NADPH. And those molecules are used in the dark reactions that are occurring still in the chloroplast in the solution around this membrane for fixing CO2 into sugars. Now, these two parts of the reaction, they work in series. One is using what the other is producing. And then, there is NADP plus have to be back, being an acceptor of the electron for the light reaction and so on. The point is that these are two different types of processes, and they are then influenced differently from the environment. Let's say that it is a very sunny spring day. There is a lot of photons around. Temperature is a bit low. what is happening there is that the light reaction in principle can go very fast. They absorb all this photon and can produce a lot of ATP and LPH, but the dark reaction are an enzymatic reaction. The enzymes depend very much on temperature, and they cannot speed up that much. So at a certain point, this is not able to really to follow, and then it stops. The excitation energy will remain in this membrane, so the molecules are excited, but the energy cannot be used for photochemistry anymore. There is a high probability for the chlorophylls for intersystem crossing. They make triplets. Or in other way, they create radical oxygen species that can kill everything. And this is super dangerous, of course. I always to say, yeah, the plants are risking their life to make food for us. And because this is a very risky thing, they have developed a series of mechanisms to avoid, because if something like that happens, they are dead. And I don't know how green fingers you are, but most of the plants are quite resistant, and simply because they have a lot of mechanisms to protect them. And so this also means that photosynthesis works in a very different way in different light regime. In this case, where there is very little light, the quantum efficiency is very high, like I told you before, almost 100%. Instead, in this condition, when there is a lot of light, large part of the energy that is absorbed by the system is actually dissipated as heat to avoid damage. And this large part can be up to 90%. So really, then the quantum efficiency goes extremely down. And the very bad thing is that, of course, light is a variable form of energy and can be very, very quickly if there is a wind, if there is a cloud in front of a plant. So plants need to switch on and off protection mechanisms very quickly to be able to do photosynthesis when there is very little light, and to be photoprotected when there is too much light. And they are actually quite good in doing that, although maybe can gain a little bit more efficiency. Okay, so here is, for example, what is happening. This is the rate of photosynthesis. This is light intensity. At the beginning, more photons. Of course, light is increasing the direction. More photons, more photosynthesis makes sense. At a certain point, everything plateaus, and the light is increasing, but the photosynthesis is not increasing anymore because large part of the energy is just discarded in a process that are called non-photochemical quenching. That is what I'm going to talk in the first part. So the question here is, where is this energy gone? So first, how do we measure that photosynthesis? How do we measure the state of the plant? How do we measure this quenching? And we do it with fluorescence. And this is a super easy scheme. So when a chlorophyll gets excited, get a photon, goes to the excited state, and then it can, the energy needs to be used hopefully for photosynthesis most of the time when photosynthesis is on, but there are also the other decay processes, that is heat, then is fluorescence, or this is an intersystem crossing. At the moment that you switch off photosynthesis, of course, all the other decay the efficiency of the other decays increases. And because fluorescence is just the emission of photons, that is something that we can measure, we can simply monitor the amount of fluorescence coming out from our leaf to identify the status of our photosynthetic apparatus and the status of plants. In fact, you can do experiment using different pulses of light in a way that you are asking a question to your plant, and the plant responds just re-emitting light with fluorescence. We can do this in steady state. So this is the quantum efficiency of fluorescence. Of course, it changes depending on the processes that are on. We can do this also resolve in time, and this is what we do a lot in my lab. So what we have with the laser, we excite our sample, and the sample can be a leaf. In this rotating cuvette, as you see in a moment, there are actually leaves that are going around. We excite the leaf with a very short laser pulse, and then we collect the fluorescence. And this is now time in nanoseconds. This is fluorescence intensity, and you see that is decaying with a certain lifetime. And this is when the photochemistry is on, because this, I don't know if you see it, here is our little laser. So really very little amount of light just to excite the system. We can also give a far more light, what we call a saturating pulse. So we are closing all the reaction centers. Means we are just closing photochemistry. Now, all the photosystems in the leaf, or at least in this part of the leaf, they have given an electron already away, so they cannot do photochemistry anymore. We can measure fluorescence again, and now you can see the decay far slower, because, of course, there is no competition for photochemistry anymore. Then we can pretend that there is a highlight, and we switch on the sun, and this is a highlight situation. We still keep the saturating pulse, so we still keep the photochemistry close, off, and then you can see that now, instead of there is the decay, is far faster than when there was no sun around. And this is simply because the plants have switched on the protoprotection mechanism. So it's throwing away, it's now introducing a new decay mechanism that is throwing away a large part of the energy that it was absorbed. And actually, we can play around with this instrument, and recently we were able even to make some measurements at low temperature. We were interested to study spruce and all these Nordic trees that are evergreens, because of course, very low temperature, you cannot do any chemistry there, and still they are green. How do they manage? And then we did this because we could really cool down everything at -20. The plants were arriving at -20 from Umeå in the north of Sweden, and they were just put in our machine, and then we could make a nice paper and also a nice card for Christmas, actually, because you can really measure in the needles the photosynthesis that is occurring there. Okay, so this is how we measure this, but the first question is, where is this quenching occur? Where is this energy disappearing? There have been a lot of discussion in the literature in the last 20 years or so. This is, again, the structure of photosystem 2 with all the different components. I told you before there are many proteins, and people have been discussing for many years, is this, the quenching is here, the quenching is here, the quenching is here, and so on. And why was it so difficult? Because actually, there are many genes encoding for all these proteins. And for example, this LAC2, I told you, the most abundant membrane protein in nature, of course, only in a little plant like Arabidopsis thaliana, that is our model plant, there are 9 different genes that are encoding for this protein. So until relatively recently, it was very difficult to knock out them all. And now, of course, with gene editing, CRISPR-Cas, you're all familiar with this, all of these things are becoming feasible. And this is actually what we did. So this is Arabidopsis thaliana. It's actually a quite ugly plant that if you have in your garden, you probably try to get rid of. But it has been the model plant for many years because it was the first one, the genome of which was sequenced. And many mutants were produced. So this is a model plant for fire plant. So we can have a wild-type system in which we have a photosystem 2 with all the different proteins, all the different subunits. We can have a mutant that is missing, and you see it's missing the big red one, green one here. And we can even have a mutant that is missing all the colorful one, and there's only the core, and the core is where photochemistry occurs. And then we can do all this experiment, measure time-resolved fluorescence, and see what is happening and compare these different plants, doing everything on plants, on leaf, without purifying actually anything. And I will make the story very short. You also, of course, need to do some modeling because not everything is so simple. But the answer to the question is that a large part of the energy disappears, so the quenching site is in LAC2, although other components of photosystem 2, they seem also to participate. So at the moment that LAC2 is not there, there is a little quenching that is associated with all these other proteins. So it seems that they all have some capacity to quench the energy or to make this energy disappear. Next question is, how does this quenching occur? And before this, let me introduce a little bit better LAC2, my favorite protein. Now you see it here in a monomer. So it's a relatively small protein. It's fully embedded in the membrane. So these are 3 transmembrane helices. And as you see, contains a lot of pigments, chlorophyll and carotenoids. So it's really packed. Some people say it's a bag of pigments. It's really packed with pigments because they are the ones that are absorbing light. So these are the absorption spectra of 2 types of chlorophyll that are inside LAC2. This is chlorophyll A, this is chlorophyll B. Chemically, it's a methyl for a 4-mil, so they are very similar molecules. And yeah, that's, their absorption is slightly different. But when this same, chemically same, exactly the same, in the same ratio, chlorophyll are embedded in LAC2, then you start to see a lot of variation in the absorption. And this is because now every of the pigment is in a different environment in a way. So, and then this tunes their absorption property. Not only tunes their absorption property, but tunes their spectroscopical property more in general. And I show you here, this is a single molecule experiment. So what you have here on the, oops, sorry. Let's see if I can go back. What you have here is a single molecule experiment. You have 1 molecule of LAC2 under a microscope, and you measure the fluorescence coming out of it. So this is a wavelength, and this is time in seconds. And this is, if you cut like this, you see the spectrum, and the spectrum is showing in this video here. And you can see the spectrum is really jumping from emission around 680 to emission about 700 nanometer, then disappearing, then reappearing again, and so on. And all of this is due to the fact that the protein is not static. Actually, the protein is very dynamic. These are molecular dynamic simulation of this LAC2, where you really see that the pigments are changing. When the pigments are changing, their environment is changing, their coupling is changing, and this tunes their properties. So these proteins, they exist in different conformations. This is what this experiment tells you. And how big this conformational change needs to be to see, for example, a difference between, so a jump in emission, this is fluorescence emission, from 680 to 710 nanometers. So many years ago, we actually did an experiment. We took one of these proteins that have a very red emission normally, or 730 nanometers here. And this is just zooming in. These are 2 chlorophylls that are connected to the protein via 2 ligands. And what we did, and they are the responsible for this red emission, what we did, we simply changed one of these ligands from an asparagine with a glutamine. We put is 1 CC bond more, so 1.3 angstrom difference. We are pushing away 1 chlorophyll from the other, and then we get a blue fluorescence. So this means that very small changes in the distance between pigment, in the arrangement with pigment, can really change drastically the spectroscopical properties of our complex. So the idea at the moment is that this LAC2 exists in 2 conformations. One conformation is a light harvesting one. It's characterized by very long excited state lifetime because it's acting as an antenna, so it has to have time to deliver the energy to the reaction center where it can be used for photochemistry. And the other instead is a quench conformation, which is characterized by a very short excited state lifetime because there a new mechanism enters and taking away all the energy. And yeah, it's just moving between these 2 conformations. Now, what is the quenching mechanism? You want to know what is the physics mechanism behind this, where this real energy goes. And these are also been a very difficult question to answer because, of course, to look for dark state in the plant is not very easy, so practically impossible. So we always have to purify the protein. But once we were purifying the proteins, then they were not quenched anymore. And so then we could not study the quench conformation. But then we got very lucky, and we can call it serendipity. A few years ago, I was actually seeing a colleague of mine showing this beautiful tobacco plant that are orange. And I said, wow, that is wonderful. I have to take it back to the Netherlands. It's really our... And this is orange because what he was trying to do is a biotechnologist. He was trying to make astaxanthin. Astaxanthin is this carotenoid here. and it's for sure very familiar to you because it's the carotenoid that gives the color to salmon and garnalen and all these pinky fishes-like things. And it's very costly because, of course, the industry, the fishery industry, want to give it as a supplement to the food to make our salmon nicely pink. So what he did, he overexpressed genes that are making this astaxanthin. This astaxanthin is never present in plants. It's not a carotenoid that plants know. But as you can see, they can grow and become extremely orange, even make a huge amount of this. So I was studying this for completely other reasons, but then we happened to purify LAC2 from these plants, and actually it was quenched. This is the normal lifetime of the excited state of LAC2, 3.5 nanoseconds, so very long lifetime because we want to have the time to transfer the energy to the reaction center. And then when instead there is astaxanthin inside, the lifetime goes to 800 picoseconds, strongly reduced. So then we had in our hand now a complex that was in a monomeric state, and we could study by transit absorption. So what we do now, we do spectroscopy, but instead of looking for fluorescence, we look at changes in absorption. So we excite with this is a pump probe technique. We excite, we pump our system in the excited state, and then with a certain delay, we register an absorption spectrum. Pump, 10 femtoseconds after absorption spectrum. Pump again, after 20 seconds, another absorption spectrum, and so on. And from the difference of the spectra, pump, 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 we can see where the energy is. And also this, I will make the story pretty short. There are a lot of components there, but we started to see that there is a new component appearing in this particular complex in around 2 picoseconds that was not present in the unquenched complexes. And the story now goes in this way. There are, I told you there are chlorophylls and there are also carotenoids there. And this carotenoid, that now is astaxanthin, exists in different states. And some of these states, they act as a light harvesting. So the carotenoid absorb very strongly in the visible. So they can absorb photons and transfer it to the chlorophyll and then to the reaction center for the photochemistry. But other carotenoids, they can act the other way around. They take energy for a chlorophyll and they dissipate it as heat very quickly. And carotenoids are perfect quenchers, so are perfect molecule to make energy disappear because the excited state lifetime of the first excited state is very short, is in picoseconds. So this means that when they are excited, they decay very quickly. They dissipate the energy as heat immediately. For a chlorophyll, it takes 5 nanoseconds. For a carotenoid, it takes 10, 15 picoseconds. So if the energy is there, it's gone. And the important thing was that we had astaxanthin in all the binding sites. We didn't have different carotenoids. It was exactly the same molecule, chemical molecules. So how come that in one case is a donor, in the other case is an acceptor? And this is our explanation. So it's a possibility that there are rotation around the ring. This is a very long chain of conjugated double bonds. And then there are a rotation of the rings on top of the bottom. This changes also the conjugation length. They can change the coupling with other chlorophyll. And then it creates a new states that can act as a quencher. Now, is it true that there are this possibility to turn around the bond in the ring? We went back to our molecular dynamic simulations, and actually it seems to happen. Of course, this was a system with astaxanthin, a very special one. What is not a natural system? What is happening in a natural system? And I told you before that natural systems are a bit more difficult to study, but at a certain point I had a very brave PhD student that decided really to go into it. And he took one other of the pigment protein complex of the family of LACs called CP29, very similar to LAC2. And in this case, it was possible to purify it in a mixed population. We have something like 80% of this protein was in a light harvested state and 20% in a quenched state. He did the same experiment, a lot of also data analysis, and he found out that actually the process was exactly the same. And then, because this is a natural system, we could also go back to the plant and make single point mutation in this protein in the plant. For example, we can take out this chlorophyll or this chlorophyll. We change the ligand, we modify the gene, we put the modified gene in the plant, and the plant make our mutant. And then we can check what is happening, and then it comes out that not really this, but this part was the one that it was responsible for the quenching. So this carotenoid with close to this chlorophyll. So this is the status for plant for the moment. But plants are not the only organisms doing oxygenic photosynthesis. There are algae, there are eukaryotic systems, but there are also prokaryotic systems like the cyanobacteria, that they are the ancestors of our plant. Cyanobacteria, they do photosynthesis in a very similar way than plants, but for the antenna system. For this, they don't have this LAC2 that I've been talking about to now, but they have a different system for collecting light. They are called phycobilisomes. So this is again now photosystem 2, and you may remember my LACs were right and left of this core, and now instead the antenna is on top of a membrane. And this antenna is different from the one of plant. It doesn't contain chlorophylls, contain bilins. Bilins are open tetrapyroles, and they absorb in the green because, yeah, you know, they are living in water, so the penetration of red light in water is not very good. So they mainly need to absorb green and blue light. And these are enormous systems containing different types of pigments. And it has been shown by others that there is one protein that is necessary for the quenching. It's called OCP, orange carotenoid protein. It also contains the carotenoids. And then this orange carotenoid protein only contains one carotenoid and is associated with this big phycobilisome when quenching is needed. And I guess you can guess the end of the story here. We did the same experiment with this system, and we actually found exactly the same mechanism. So different components, different proteins, different pigments, bilins instead of chlorophylls, but physically the same mechanism. Energy transfer to a carotenoid, and then is dissipated energy very quickly. Okay, this was my first part. Shall I stop here, or I go a bit, 5 minutes more?
No, 5 minutes.
Yeah? And because my second part was the highlight, and my second part is the photosynthesis in the shade and infrared light photosynthesis. And I will just give you an introduction, and then we'll explain later what we are doing there. Now, well, this is familiar to all of you, is the spectrum of the sun, and the yellow one is the spectrum on top of the atmosphere, and the colorful one is the spectrum at sea level. Photosynthetic organisms, the one doing oxygenic photosynthesis, so using water as an electron donor, they are using only part of the spectrum, that is up to 700 nanometers, and we are we have a lot of visible photons, and of course visible photons are more energetic than the others, so it makes very much sense. However, not all the rest is all discarded, and of course we cannot use, not all these photons can be used because some of them, or a large part of them, they don't carry enough energy to take electrons out of water, but one part of them, actually they do. All the photons between 700 and 800, in principle, they carry enough energy for water splitting. So why are they not used? And this has been a discussion also for a very long time. People will say, oh, you need overpotential, it's not possible, thermodynamically will. But then 10 years ago, the group of Don Bryant at Penn State University discovered some cyanobacteria that are actually able to do far red light photosynthesis. So these cyanobacteria are, if you grow them in white light, they are normal one, like the one we have always seen. But if we grow them in far red light, then they start to be able to use it. And they are able to use it because they have a gene cluster of 21 genes that are expressed when the organism is put in far red light. And what are these genes? These genes are one very important one. This one is an enzyme that is transforming a chlorophyll A, the typical chlorophyll that you have seen up to now, into a different type of chlorophyll that is called chlorophyll F. This is the absorption spectrum of chlorophyll A. You see that the lowest energy transition, so the red band has a maximum of 664 nanometers. And for chlorophyll F, in solution, not in the protein, is already above 700 nanometer. And this you get it with the substitution for a methyl for a 4-mil group. Yeah, so chemically is one reaction, one enzyme, that is this one, is making a new chlorophyll. And then the other genes are for proteins that are paralogs of the normal protein of photosystem 1 and photosystem 2 that can host this far red absorbing chlorophyll F. And then I was extremely puzzled, and I wanted to understand how do the chlorophyll F photosystem work, because of course chlorophyll F is a lower energy than a chlorophyll A. You are losing quite a lot. And this is what I'm going to tell you after the break. Yeah, welcome back. Yeah, so I left before the break talking about the fact that there are some organisms doing oxygenic photosynthesis. They can use far red light, so the photons are low in energy. And yeah, I was very interested to understand how they do that. And the first thing that we did is actually to grow this bacteria in two different light, in white light, where they are normal, and infrared light, where they become able to do photosynthesis with these low energy photons. And then we started to purify the different components of the photosynthetic apparatus. For example, the typical, as I told you before, photosystem 2 and photosystem 1. These are the absorption spectra of the purified complexes, so we take them out of the membrane, and this is the absorption, so wavelength with the intensity. And the color code is always the same. We have the black one are from organisms that are grown in white light, and the red one from organisms grown in far red light. So the spectra here, these two spectra are very similar, as you see. The main absorption is here, around 780 nanometers for both of them. But the far red one has also a shoulder above 700 nanometers. So here, this is the absorption in the far red, and this is the only difference. And the same for photosystem 1. Also there are, most of the absorption is here, and there is a little bit more here, that is the far red. And this far red here is where this chlorophyll F, this new synthesized chlorophyll, actually absorbed and is integrated in photosystem 1 and photosystem 2. Now we can also look at the fluorescence. So we look how the energy is distributed if the chlorophyll F are connected with the other chlorophylls in the system. And yeah, you have to believe me. So in the case of chlorophyll A, you see a peak here. In the case of chlorophyll F, you see only a peak here. So this means that they're all very well connected. But we can also do better. We can do a time-resolved fluorescence. So we can look at the fluorescence resolving time. And we can really look at the energy going out of a chlorophyll in another one, and how long it takes to reach the reaction center, so the photochemical center, where it can do charge separation. And this is, a measurement. Here you have a wavelength, here you have a time, and the color here are the intensity of fluorescence. Red is I. So if I cut it like this, is what I showed you before, is the decay of the fluorescence, the decay in time. And then we can take these maps and analyze them as a sum of exponential decays that are characterized by a time and a spectrum. And what we see is actually this spectra like this. I'm getting a bit technical, sorry for this, but I want to show you that we can really see the energy flowing out of this a chlorophyll absorbing here, to flowing in a chlorophyll absorbing here, out from here, in 5 picoseconds, 18 picoseconds, and so on. And then we have this green spectrum that is all positive, and this simply the energy disappears. It shows that the energy is gone because there is no negative part, and it's gone because it's used for the photochemistry. And it's take under 36 picoseconds. And this is photosystem 1 in the far red. So what we observe with this kind of measurement is really the energy, in a way, in this case, hopping from one pigment to the other, to another pigment, until it's trapped in the photochemical center, until you get an electrons out of the system. And if we look at all of this and we do this time resolve, we can then measure how long it takes from the energy from when it's absorbed by one of these pigments to when it gets trapped, what is used for the photochemistry, when you get an electron out of it. And as I told you before, this time is very important. The shorter is this time, more efficient is the process. So then, I will not go in detail there, but this photosystem 2, from our measurement, gives an efficiency of 65 to 70%. So out of 100 photons absorbed by the system, 70 of them, they lead to charge separation. For photosystem 1, it's 93. And if you remember what I told you before, for photosystem 1 in a normal photosystem was 98, 99%, and for photosystem 2 was 92%. So what is the take-home message here? Chlorophyll F is integrated in the system, but they are less efficient. So it takes longer to use this energy, and then the efficiency go down a bit. On the other hand, they can still absorb this far-red photon that before could not be used. And maybe you want to know why it's less efficient, and this is, you can show it here. This is an energy landscape. These are all the chlorophyll in the structure, and this is the energy of this chlorophyll. In a normal photosystem, all these chlorophyll are isoenergetic, so they have the same energy. It's a very flat energy landscape. Instead, when you have a chlorophyll F are far lower in energy than a chlorophyll A. So what is happening, the energy has to go from here to here, and now from here to here, it has to go down and up again, down and up again, and the up step is very slow. Yeah, for the chemists and physicists among you, it's just Boltzmann here. So this takes longer because the energy is kind of trapped in these lower energy forms. And this is slowing down the whole process, both in photosystem 2 and in photosystem 1. But photosystem 1 is so packed of chlorophyll, and there are so many ways to transfer the energy to the photochemical center that at the end, the penalty is not very big, and photosystem 2 instead, the quantum efficiency goes down 20–30%. Okay, so in principle, it seems to be good, but not perfect. In reality, if we go back to the organism, we see a spectrum like this. This is the absorption spectrum of the cyanobacteria, not of the purified things. And then you can see that the absorption here above 700 nanometers is far stronger than in the purified complexes. What is happening there? Well, actually, the major absorber for far-red photons are not the chlorophyll F in the photosystems, but are some pigments here in this phycobilizome that I introduced before, this larger antenna on top of the membrane, that actually in this far-red system, they absorb a 712 nanometer. And there are so many of those, and they are major responsible for this absorption. And then there is something very peculiar, because they are also absorbing the green, so we can excite them differently from the photosystems. And then what we observe is that when we excite them directly, the energy transfer to the photosystem 2 is far faster than when we excite directly the photosystem 2, which is very strange. Because you can well imagine that more chlorophyll I have, the larger is my antenna, the longer it will take to get the energy to the reaction center. It has to go more steps, and there are more pigments and so on. And here instead is exactly the other way around, which is a unique case because it has never been seen before in any other system. Then, how it works. Then they solve the structure of this photosystem 2, and they show where the chlorophyll F are located. One is here, and two are here. We can do some calculations, and we can calculate how long it takes for the energy to arrive here. And we discover that from this side, it takes very long time, but from this side, instead, it's very quick. So the interpretation of all our results is that this big antenna on top of the photosystem is connected via the fast highway and not the little street here. So when you excite here, the energy flows to the photochemical center very quickly. And this is why you see higher efficiency also. So it's a very nice and interesting design. Now, these days, we are discovering more and more different types of cyanobacteria that are actually able to do this far-red photosynthesis. There are many, many different gene compositions. Some have only genes for photosystem 1, some only genes for photosystem 2 with the far-red capacity, and so on. So we are really studying at the moment the different strategies for using far-red photons for photosynthesis. These are all of them, and maybe next time. But the next question is, can we use this information for also making plants or crops to do far-red photosynthesis? And maybe a a more relevant question for the moment is why should we do that? First of all, maybe this is also a surprise for you for a process that is sustaining virtually all life on our planet, 8 billion people or so on. Photosynthesis is extremely inefficient. The whole process is able to store in the form of carbohydrates less than 1% of the sunlight that is arriving at the plant site. So it's really super inefficient, and still it's giving food of all of us. And it's super inefficient because it's a very complex process made of many different steps. And every step is a loss of energy. And you can also see it in a different way. For me, photosynthesis is actually an energy transformation process. It's transforming a form of energy that is very difficult to catch and store, like light, photons, into a form of energy that can be stored forever. And every time you want to store something, you pay a price. And this is also nature, the price of nature. And the price is energy. You store less than 1% of what is actually available. So because we have to improve crop productivity, so there are a lot of research now going on to try to improve photosynthesis. You get a double efficiency, and it's a huge effect. And here are many different steps with different losses, but the first loss is at the top. You lose already 50% of the energy simply because you cannot absorb, or photosynthetic organisms cannot use photons differently from the visible photon. So all the rest is discarded, as I told you before. However, far-red photon in principle can still be used, and cyanobacteria are using them. And actually, the situation is even more interesting for crops, because now what we have here is the spectrum of the sun on top of a canopy, is the yellow one. When it's the leaf on top of this crop canopy, this crop field, get all the visible photons for around the 700 there. And then this green is the spectrum at the bottom of the canopy. And I don't know, I guess you probably have experienced if you enter in a maize field when the maize is growing, it's kind of dark down there. And because all the photons, or visible photons that you are seeing, are absorbed by the upper leaves, and there is almost no light there. And no light means no visible light, but there is a lot of far-red light because they don't absorb it. So this means that the leaf at the bottom, they do not do photosynthesis. And recently, we have also calculated this. This is the result of some modeling of a soybean canopy, in which what you have here in different colors, so this is the visible light, how it's absorbed, and what is the photosynthetic rate of the different leaves. And you have a very high photosynthetic rate at the top, and bottom, 0, simply because there are no photons arriving there. The lower leaf in this canopy cannot do photosynthesis. Now, let's switch on the capacity for this bottom leaf to do photosynthesis with the far-red photons, and then you get immediately an up to 26% increase in the photosynthetic rate if we can use this far-red photon. So there is clearly a scope to do that, and then the question is, how do we do that? And maybe the first question that you want to answer is, okay, but cyanobacteria can use far-red light. What about plants? Can they also use far-red light? And then I tried. We did the experiment. We took our little Arabidopsis thaliana. We put it in normal white light, in far-red light, and we put it also in dark because in dark, photosynthesis starts to switch off. Plants, they go to senescence. Don't know, when you put a tent in your camping and then you take it out after a week, there is all the grass is bleached, no, because photosynthesis, the chlorophyll are not synthesized anymore. So we want to discriminate between not seeing any light to see far-red photons. First of all, we also look at the pigments and say maybe we get some chlorophyll F there or some red absorbing pigments there. No, this is not the case. Arabidopsis doesn't make far-red chlorophylls. But on the other end, it's trying really its best to absorb far-red light. So because one of the two photosystems has this absorption spectrum a little bit more to the far red, what it does, it changes drastically the ratio between photosystem 1 and photosystem 2. It's making a huge antenna to try to absorb every single photon that is passing by. It's really fighting very hard to try to do photosynthesis. At the end, it doesn't manage, but at least is able to respond to this change in light, and which is a good news for us, because at the moment that I will be able to make the plant to use the far-red photons, then I don't have to adjust anything else. The plant will do it by itself. Yeah, I just... now, first question is, can we integrate chlorophyll F, this low-energy chlorophyll, in normal photosystems? So the white light photosystem, I will call. And the answer is yes. Actually, we have done this. We have taken a cyanobacteria that is not able to synthesize chlorophyll F. We have put in the enzyme, the gene for the enzyme that encode for a chlorophyll F synthase. And then this is able actually to make chlorophyll F. And this chlorophyll F is integrated in the photosystem. This is the fluorescence spectrum of photosystem 1. And this 725 is the emission that corresponds to chlorophyll F. So it's there, and it's actually connected. We've done all the spectroscopy. But now I want you to compare it with the red one. This is the emission spectrum of the photosystem 1 from far red light cyanobacteria. And you see, it's not 725, it's 790. So the answer is yes, they integrate chlorophyll F in the white light photosystems, but they don't shift it that much. So then we get up to 730 or so, and not to 800 nanometers. If we want 800 nanometers, we have to modify also the protein, because as I mentioned before, the protein is tuning the properties of the pigment. And then we started to do that. And actually, we used for this, again, our LAC2. LAC2 is a great protein, not only because it's abundant, it's modular. So when a plant is growing in high light, it makes very few of this LAC2, no? Because there are a lot of photons available. It doesn't need to increase the absorption cross-section too much. When the plant grows in low light, they make many of them. So this is really modular, and it's easy to manipulate. And the other great capacity of this LAC2 is it can refold in vitro. So you take the apoprotein, you make you express it in Escherichia coli, so in a bacterium, you have the apoprotein, you add the pigment, and magically enough, the protein refold. So you assume the native conformation with the spectroscopic properties that are identical to the native complex. And this allows you to do a lot of experiment in an easy way, because you don't have to make mutation in a plant, but you can just make mutation in a single gene, and then you have it expressed in this bacterium. So you take the apoprotein, you take pigments that we purify for plant or with cyanobacteria, we mix them, and we obtain refold the complex, and this is the absorption spectrum, so wavelength absorption. And this, you have to believe me, is identical to the complex to the spectrum of the complex that we purify from plant. But now, in plants, these are these two, chlorophyll A and chlorophyll B. Now, what if instead of chlorophyll A, we use a chlorophyll D, that is one other of this low energy chlorophyll? Well, it's also refolding. And now, instead of having a peak here at 670, as a peak of almost 700 nanometer, but it's a hole here. This is chlorophyll B, this is chlorophyll D. Then we put back a chlorophyll A, and we fill also the gap. It's also taking the three of them. And what about chlorophyll F? I was speaking about that for a long time today, and it's also taking this and is arriving up to 740 nanometer. So this complex is able to accommodate this different type of chlorophyll. So the binding site for the pigment in this type of complex are actually promiscuous, which is a very good news, because then we can identify which one are having higher affinity and study them. And actually recently, my master student did a beautiful experiment in which she was changing the ratio between the different chlorophyll, and then you can see the spectrum was also changing. So most of the binding site can accommodate both chlorophyll A and chlorophyll D, depending only how much you give during the process of refolding in vitro. Then, yeah, we did a lot of spectroscopy. I will not spend time here, just to tell you that these pigments are functionally inserted in our complexes. So are there, they absorb light and transfer excitation energy. And yeah, they are beautiful, but maybe another time. Now, where are they inserted? Which binding site? And now we know the structure, we know everything of this protein. And as I told you, we can also modify this protein. And for example, we can make mutant in which these chlorophyll are not present because we change the ligand. And these are the spectrum of the wild type, and this is the spectrum of the mutant. And you can immediately see that in these two binding sites, there was chlorophyll D. So these are. And now, from there, we know they are chlorophyll D, and then we also can do some calculations here. We can do some molecular dynamic simulation in which we change the binding site. Instead of having chlorophyll A or B, we can put chlorophyll D or F, and we run molecular dynamic simulations. We take a different picture of this, the snapshot of the molecular dynamic simulation, and we do calculations in which we want to see, yeah, let's say that in this side there is a chlorophyll A or is a chlorophyll D, which amino acid are facilitating the binding of one type of chlorophyll or the other type of chlorophyll, and what is the energy of the binding. So we can have all of this. We map the full LAC2, and then we can, yeah, fundamentally determine what are the rules for having binding sites that are accommodating one type of chlorophyll or the other. And then next is then we want to now optimize this kind of complex and put it back in vivo in the plant together with the chlorophyll F synthase. It will take me a few years to get there, but this is the direction in which we are going. And actually, very last slide for this part, I told you that the protein is also able to shift, to tune the absorption even more to the far red. And actually, recently, we managed to make a protein in which this chlorophyll F is above 750 nanometers. This is because we are inducing some strong excitonic interaction between pigments. Yeah, so the pigments are very close together. They feel each other. So using proteins and pigments, then we really can more or less exploit large part of the far red light. Now, next step is again to put it in plant and maybe see you in 5 years. And with this, I would just like to thank all the people who did this work. So there are many people passing by my lab that they've given a great contribution, and of course also many different collaborators for different parts, and also in Groningen, we started to do the molecular dynamic simulation thanks to my PhD student Nicoletta Liguori, and with, of course, the help of Sivarian Marlik. And thank you very much for your attention.
Thank you very much, Roberta, for this fascinating walk in a very complex world of photosynthesis. I think we have all learned something tonight, but for sure, I'm convinced there are also some questions. Ton?
Most interesting story. I was reminded of the algae that are in the deep sea. In the deep sea, I assume that there will be more infrared light down in the deep sea. So do these algae also have this kind of mechanism, or do they, let's say, have the form of chemical autotrophic?
So in the deep sea, you are meaning with the vent, and so there is some far red light. Of course, there is no penetration of far red light in the deep sea, but for this vent, they are creating some far red light. And there are some organisms that are doing anoxygenic photosynthesis. So for example, purple bacteria or sulfur bacteria, they do not use water as a electron donor, but they use a sulfur as an electron donor. And then they use this far red light coming from this vent mainly, yeah. But then they can go up to 1,000 nanometers in absorption, but again, they don't split water, don't take electron from water, yeah.
Over here.
So without knowing too much about the topic, I think you suggested that by using this far red light, you would be able to make a plant greener lower to the ground. So are you suggesting that you can that you can deliver more biomaterial so from a crop? And do you dare to predict how much more yield you will get in this case?
Yeah, so yes, this is exactly what I want to do. I want to make the lower part of the plant absorb far red light, not the whole plant. I want to induce the synthesis of this chlorophyll F and of the protein that are able to bind this chlorophyll F only in the lower part of the canopy. And according to our calculations, the maximum increase of yield is 26% of photosynthesis rate. In photosynthesis rate, it's biomass, it's almost direct. We can argue. But this means that everything should be perfect. And perfect means that I also don't have to be, I have to be able to absorb this light and also transfer it to the reaction center efficiently, which means that I have to know where to put this chlorophyll and how many of them. But that is the whole idea of the project. Yeah, that is where I'm aiming.
Thank you for your very interesting lecture. My question is about quenching. You mentioned carotenoids like astaxanthin and phycobilins as a quenching system. Do plants need these kinds of accessory pigments for quenching, and how important is that? Can that be an evolutionary trigger also to develop these kinds of systems, or is harvesting energy still more important than quenching for these systems?
Yeah, so the plants, they are completely dependent on carotenoids. A plant depleted in carotenoid will not survive 1 second out in the sunlight because carotenoids are not only doing this type of quenching, but also scavenging singlet oxygen. So that's no carotenoid, no plant, no food. So that is something they cannot do without, yeah.
And for this quenching system, is that also, they are they are necessary for quenching, or can chlorophylls also do the job, only less efficient?
No, so for the quenching, there are many different, I was presenting one, the fast one. There are plants that do not have this mechanism. They still survive, but they make less kits, let's put it like this. Let's do less seeds. So they are far less productive, and they get damaged more easily.
There was another question over there.
Yeah, thank you very much for this wonderful lecture. My question is, please, I guess that when the sunlight is entering a green leaf, then probably it will get polarized. And after that, maybe within those complicated molecules you show to us, it will get resonant to various extents. But what would be the positive impact of those resonances within, for example, a chloroplast or other plastide?
Yeah, that is an interesting thing. I think there are not many people that have been exploring this. A few years ago, we did a work with somebody that was interested in photonics. I was a professor at the University of Amsterdam. And the idea was that if you look at this first structure that I was showing in the membrane, these pillars of membranes, they look like they can be at the right distance for enhancing effects. But yeah, there are a few groups working on that, but I don't really have a clear answer. It seems that there is a bit of an answer, but we'll see. Yeah, it depends on the distance between those. And,
There was one question there, and then Wim.
So you talked about the 26% yield, you wanted to have the F from 730 nanometers to about 800 nanometers. So is the 26% yield for the 800 nanometers, or what is the yield for 730 nanometers?
Yeah, so this is for 760. These are the data that we got for 760, up to 80%, absorbing up to 80%, if I'm not, if I remember correctly, up to 760. Yeah. Though I don't know how to. We didn't calculate for 730, but it would be less. Yeah, probably 13 or something like this. But again, this is really the maximum. I had to transport everything efficiently to the reaction center. Still. It's an interesting system.
Thank you, Roberta. I learned a lot. I was puzzled by your funnel that you showed. Photosynthesis is not very efficient. You showed it's only 1%. But in the top, that's what your work is, it is quite efficient. About 50% is still converted. From a biotechnology viewpoint of view, shouldn't you work on the lower part?
Yeah, I'm in the laser lab, so I guess no, yeah. So there are people working on the different parts. So one of the major losses occurs with this non-photochemical quenching, for example. There are quite some papers in the last few years that are showing that this plant, so in low light they are very efficient, but they are exposed to fluctuating light. So what they do, they switch on the quenching mechanism very quickly when they see high light, and it takes quite some time to switch them off, because fundamentally, yeah, this is my interpretation of the psychology of a plant, they want to play safe. No, then they want to be prepared and then in that case, they are still throwing away energy when actually they are already in light limiting condition. And there have been some papers showing that, for example, it was the way of tobacco and also soybean, that they can have up to 33% increase in biomass if this mechanism of switching is faster. On the other hand, they did the same in potato and Arabidopsis, they didn't see anything. So there's still we needed to understand something there. So that is the part of the light reaction, and then, of course, the absorption. And the other thing that can also be done is to make the plants a little bit more transparent, meaning that at the moment what we have all the leaf on top that they have too much light, they have to throw away when there is too much light, and the leaf on the bottom, they don't get anything. If we make the plants a little bit more transparent, so less chlorophyll at all levels, then you have a better penetration in the canopy, and this is also another direction in which we are actually working. So that's, and then the other part is, of course, the dark reactions, and the dark reaction, the big problem is rubisco. Rubisco is the enzyme that is doing the first step of CO2 transformation to carbohydrate that likes oxygen a bit too much, and so people are also working in many of these parts, but yeah, that's his different department.
Okay, I think we put the rest to the oral session of the questions, and I would like to ask you to join me again, thanking Roberta for this very interesting talk.
Thank you.
And of course, also we have our little souvenir book for you, which I hope you will like.
Yeah, for sure. Thank you.
And for the next time, as you can see on our board there. Oh, yeah. Sorry. Ik was nog aan het lezen tegenover, maar het staat ook hier. Op dinsdag 11 maart hebben wij Marie-José van Tol, die ons over depressie en fantasieren zal toespreken. Ik hoop jullie allemaal weer terug te zien komende maand. Dank u wel.